Eukaryotic meiotic recombination occurs between replicated non-sister parental homologs. This means that the two chromosomes, one maternal and one paternal, have the same gene sequence, but the alleles may differ. When DSBs occur, homologous recombination is induced, which occurs due to a template strand being present. Recombination is a crossover event in which segments from one chromosome change to the other chromosome. This is important to increase genetic diversity. SPO11 is a gene that codes for the SPO11 cleaving protein. SPO11 protein attacks and uses tyrosine to cut the phosphodiester backbone. Specifically, the SPO11 subunits break apart and each bind to the 5' end of the single strands. The phosphodiester bond is cleaved 2 nucleotides apart in the DNA so that the DSB is slightly staggered. SPO11 remains bound to the DNA and recruits other factors. One of these factors is the MRX complex. The MRX complex is composed of the subunits MRE11, XRS2, and RAD50 that are encoded by the corresponding genes. It is recruited to the DSB and binds to the 5' end on each single strand at the site of SPO11. SPO11 falls off. The MRX complexes degrade DNA in the 5' to 3' direction, generating 3' overhangs that can be as long as 1,000 bases. The RAD52, RAD51, and DAMC1 proteins are recruited to the DSB to facilitate strand invasion. RAD51 is made active through interaction with RAD52. Together, these 3 proteins bind to the 3' ends of both single DNA strands. RAD52 falls off, leaving RAD51 and DAMC1 to facilitate strand invasion, just like the prokaryotic homolog REC-A. Once strand invasion is complete, DNA polymerase is recruited, binds to the 3' ends of the ssDNA, and synthesizes the new DNA in the 5 to 3' direction. Once the new DNA is made, a structure called the holiday junction is formed. The holiday junction is resolved in one of 2 ways. The rare case involves the resolvase, either SLX1, SLX4, or MUS8, 1MSM4, that cleaves at site 2 only. This leads to a patched, non-crossover event. The most common method, which is shown here, involves resolvase cleavage at both sites 1 and 2, resulting in crossover products.